And thank you, Arnaud Dubois, for already making the bridge to Raman spectroscopy. So a brief overview over what I'm going to be talking about. I will very briefly introduce Raman spectroscopy and then show in not too much detail, but sufficient detail, I hope, what it gives in terms of insights into what I call the molecular skin anatomy. So not just the molecular composition, but also how that changes with depth into the skin. I'll do this first for in vivo skin and focus a little bit on what it can tell us about skin barrier and skin penetration of topically applied products, and then compare what we have seen throughout the years, how skin barrier and skin penetration, how that performs in skin models. And I'll finish up with a wrap up of the findings. So meet Mr. Raman, a very clever Indian scientist who discovered the light scattering effect on which Raman spectroscopy is based almost 100 years ago in 1928. So all you need to know to understand Raman spectroscopy is that the atoms in a molecule do not stand still, but they move about their equilibrium position in the molecule in so-called vibrational modes. Now, what can happen and what Mr. Raman discovered is that if a particle of light, a photon, hits a molecule, it can transfer some of its energy to such a vibrational mode. So if some of the energy stays behind in the molecule, of course, the scattered light will have less energy, and that's what you detect as a shift in wavelength. Now, the amount of energy that is needed to excite such a vibrational mode in a molecule is very tightly defined. It depends on the masses of the atoms involved, their chemical bond, and for larger molecules, also things like secondary structure and even interactions with other molecules come into play. Now, molecules can have many different vibrational modes, each with their own bit of energy that is needed to excite them. And what this results in is that if you illuminate molecules with monochromatic light, laser light, then what you get back is basically a rainbow of colors. And you see some examples here of Raman spectra. So you can interpret this wave number axis. It's actually an energy axis, but you could see it as a wavelength axis, if you like. The thing I would like you to remember is that this shows very clearly that to a high degree, Raman spectra are molecule specific. So you can identify molecules based on their Raman spectrum. Now, if you apply this to tissue, you get very complex spectra because all the molecules present in the tissue contribute their own Raman signal to this one tissue Raman spectrum. Nevertheless, a tissue Raman spectrum, in this case a skin spectrum, is a one-to-one representation of the overall molecular composition of the skin. Raman spectra are not as detailed, do not give as detailed molecular information as mass spectroscopy. Raman spectra are not as sensitive in terms of limits of detection as, for instance, an HPLC, but they have the big advantage that it's non-invasive. It doesn't require any labeling, so it can be readily applied in vivo. That's what we did. So some 20 years ago, we introduced our first in vivo skin analyzer based on Raman spectroscopy. It has evolved a little bit over the time. It's become a little bit more user-friendly also. So basically it can be used nowadays without you first having to become a Raman expert or a techie. So as you see, it's a benchtop system. You see a white support on which the skin is placed. And in the center of this white support is a window through which a microscope objective is looking up. Let me give you, so you see the microscope objective here. And this microscope objective focuses light into the skin that's resting on the window. It's a confocal system, so the microscope objective also collects Raman scattered light, but only from the position where the laser focus is, not from the areas below or above. And by now, moving the microscope objective up, you focus deeper and deeper into the skin. And in this way, you get spatially resolved Raman spectra, which tell us about the molecular composition at that position in the skin. Now, it's not just applicable to the volar forearm, although that's where most experiments are usually done, but also the axilla is accessible, the scalp, the hand, if you're flexible enough, the leg, and various locations on the face are accessible for measurements. Measurements take place in two regions of the Raman spectrum. The so-called fingerprint region, which, as you can see, has many different bands, lots of spectroscopic information, therefore molecular information. And the high wave number region, which looks a little bit boring, perhaps, but gives detailed information about, for instance, the water concentration in the tissue. So here we focus in on the high wave number region, which has signal contributions of primarily proteins and some lipid contributions and water. So the proteins and the water. Now, the nice thing of Raman spectra and the Raman intensities is that they go linear with the concentration of a molecule in the measurement volume. So that means that we can interpret the spectrum as this by looking at the intensity ratio between the protein signal and the water signal. And that gives us results such as here, which is a water concentration profile going from the skin surface into the skin. And we see that typically looks like this. At the skin surface, you have a water concentration of usually between 20 and 30%. Then it goes steeply up through the stratum corneum. And at about 65% is where you move from the stratum corneum into the living epidermis. And then if you go deeper, you see another slight increase where you move from the living epidermis into the dermis. So this is a very nice illustration of the barrier function of the skin. The fingerprint region has contributions of many different molecules, proteins, amino acids, sweat constituents such as lactate and urea, and natural moisturizing factor. So I will not go into how we extract this type of information from these spectra, but basically you can obtain information about the distribution of proteins, amino acids and their derivatives, sweat constituents, and lipids in the stratum corneum and deeper. So this has been used as we have not because I want to top Arnaud Dubois, who talked about 100 papers. This is coincidence. There's about 200 papers currently in the scientific literature where this type of information has been used to look, for instance, at the effects of UV exposure or the changing of the climate through the seasons on the distribution and the composition, molecular composition of the skin, as well as the effects of aging, the different anatomical locations, and the response of the skin to various challenges. Now, what you can do for molecules that are intrinsically present in the skin, you can also do for molecules that you apply to this, or for products that you apply to the skin and that penetrate into the skin. And this, as I said, has been described extensively in the literature. and still every year, quite a few papers are being added to this. Now, if you talk about skin penetration, so you apply something, and basically for Raman spectroscopy, the only thing that changes is if an active, for instance, penetrates into the skin, then you will not only detect the Raman signal of the intrinsic skin components, but also the Raman signal of the product penetrating into the skin. And this can be analyzed fully quantitatively. So you apply a product to the skin, you let it sit for the amount of time that you think is right, and then you start taking Raman measurements. So you take Raman profiles. You usually do this at some 10 different locations within the treated area to average out biological variance. Every profile takes about 1 minute to take, so that could take about 10 minutes then. Every Raman profile can be converted into a product concentration profile, so the concentration of the product as a function of depth into the skin. And when you take the area under the curve of such a profile, you get the total amount in the skin. And because it's a non-invasive technique, you can repeat these measurements as often as you want. And in that way, monitor how a product diffuses further into the skin. So you find less and less in the stratum corneum, close to the skin surface. And in this way, you can actually get a quantitative measure of the flux of products into the living skin. So as an example of that, very simple example, we applied a mixture of 0.3% retinol, dissolved in 70% ethanol and 30% propylene glycol, was applied to the forearm, and we let it sit for 10 minutes. The ethanol, because it was not occluded, the ethanol evaporates, and the retinol and the propylene glycol penetrate into the skin. So here you see the result for retinol. So quantitative result in milligrams per mil. The concentration of retinol immediately after stopping the application of the product, 1 hour later, etc. So we followed it for 6 hours. Taking the area under this curve, so you see a gradual decrease of the concentration, which is also reflected in the area under the curve. So the total amount in the skin, see a steady decrease. And from this, you can simply calculate a flux of the product into the skin. Now, from the same measurements, you do not only get information about the retinol, but also on the propylene glycol. And the first thing that is obvious is if you compare what happens to the retinol, where you see a gradual decrease, and you look now at what happens to the propylene glycol, you do not only see a decrease, a slight decrease, but you also see a sort of redistribution of the propylene glycol throughout the stratum corneum over time. And also here, you can use the area under the curve to calculate a flux of propylene glycol deeper into the skin. Here's another example where we applied actually a disinfection cream with a high percentage of ethanol to the skin, and we simply followed it for half an hour. So we applied it for, I think, 5 minutes, and then we simply followed what happened to the ethanol in the skin over a period of half an hour. So here you see what happens to the concentration, and by taking the first derivative of that, you get actually a measure of the flux of ethanol into the skin. So I think this is something that perhaps Yuri Danzig can put his teeth in trying to model this. So that's about in vivo applications, very short and very brief. Of course, the instrument doesn't know whether there's an arm attached to the skin that's lying on the window. So you can apply this method just as well to skin models as you can to in vivo skin. And that offers an opportunity to see how well the skin models perform in terms of their molecular anatomy when compared to the in vivo skin situation. Um, so I cannot say that we've made a very systematic study of all the skin models, but that are available, and that we probably don't have all the newest skin models and skin and developments there. But throughout the years, we've had the opportunity to do some measurements on skin models, and one of the first was on the skin model that was then available from EpiSkin. So we see here side by side in orange the in vivo skin result. This is, for instance, if you look at the top left corner, you see the distribution of natural moisturizing factor as a function of distance to the skin surface in vivo skin, and the result for the EpiSkin skin model. So you see quite a high concentration of NMF in the skin in vivo, and you see no NMF in the EpiSkin. Now, as you know, most of the natural moisturizing factor arises from the enzymatic degradation of filaggrin in the stratum corneum. And this goes to quite high concentrations. The dry weight percentage of NMF in the stratum corneum can be as high as 25%. So this is something that's easily picked up with Raman spectroscopy. But as I said, where you see a lot of NMF signal in vivo skin, there was no NMF signal in the epi-skin. Ceramides, free fatty acids, and cholesterol, especially in the early years, let me put it that way, were usually much higher in skin models than in vivo skin. And what was usually lacking was a clear water concentration profile, as I've shown you earlier for the in vivo skin. So with the very low start at the skin surface and then a steep increase. Here is another on a skin-ethic reconstructed human epidermis model from a group in Switzerland, similar to what we found for the previous skin model. They saw no. They did not find the typical water concentration profile of in vivo skin, as you see here, but instead found basically no profile at all. Also here, the NMF was lacking in the skin model, and also here, the lipids were quite a bit higher than you would find in vivo skin. What these researchers also did was to look at the penetration of various products into the skin model. So they compared in vivo and skin model for instance, caffeine, benzoic acid, and salicylic acid, where you find a clear profile in the in vivo skin, so high concentration in the stratum corneum, which of course is the actual barrier for penetration into the skin. They found a very homogeneous distribution for caffeine, benzoic acid, and salicylic acid. And for two other products, they found concentrations in the skin model that were 1 to 2 orders of magnitude higher than you would find in vivo skin. So there's some more results. This is also an interesting one. It's a very recent one where we did a test together with researchers from the Leiden University Medical Center. We developed skin organoids. So here's a comparison of in vivo skin again, and in orange, the spectrum that we obtained from the skin organoids. And we see a few very strong bands in the skin organoids that you do not find in the in vivo skin, and these are due to glycogen. And that actually makes sense because it turned out that the organoids are grown on a very high glucose medium. So the quiet conclusion is that the skin models in terms of their molecular anatomy still leave something to wish for when compared to the actual in vivo skin situation. There's very little in terms of skin barrier formation. I mean, actually functional skin barrier formation. But we also saw how, if you look at the skin organoid results, that the way how you culture something and the way how you culture the models has an effect on their molecular composition. So this can also be turned around, of course. That means that Raman spectroscopy offers a method to actually test how well your skin models resemble actual in vivo skin, and also how your culture conditions affect their composition. This has been used in a few papers in the literature, but not enough for my liking. But one of those papers is actually from Yuri Danzig, who looked at the effect of the time that you culture such a skin model, and also the effect in this case of IL-4 treatment on how these skin models develop, looking at stratum corneum thickness, etc. Now, for those of you who don't like to look at spectra, there is good news because in the last few years, quite a few companies have started developing methods and making them available, Raman imaging methods. So instead of looking at spectra, you can look at the image of a tissue sample, for instance, imaged at the wavelength of one of these Raman bands, which represent molecular composition, so real label-free chemical imaging. And even by combining the images and the contrast that you find at different wave numbers, so at different peaks, Raman peaks, by combining these, you from living and fresh, unfixated tissue, you can obtain virtual staining images that look very much like what, for instance, a pathologist looks like in H&E stain slides. And this can, and Lightcore, I mentioned Lightcore here because that's their images, but there are other companies active in this field, also in Europe, Cambridge Raman Imaging in Milano, Refined Laser Systems in Germany. And these imaging techniques, let me see if I can start. Yeah. They can also be used almost at video rate. So you see here some scans through an organoid, not a skin organoid in this case. But so you see almost like an H&E like slides, but this is fresh tissue, virtual staining. So without using any labeling or stains. Now what you can do, this is of course gives you a lot of structural information, and just as in the previous talk suggested for LCOCT, you can combine this with spontaneous Raman spectroscopy to get the full spectroscopic information at any location that you want for further interpretation. And this is something that we're starting to explore together with Lightcore Technologies in Marseille. So to wrap this up, I think the conclusion has to be, and again, we have not studied every single skin model available that's present, but skin models do seem to struggle to reproduce the in vivo molecular anatomy and the in vivo barrier function of the skin. The good news is that Raman spectroscopy and imaging offer a non-invasive label-free approach to test those skin models and to monitor what happens while culturing such skin models, and in this way to improve skin models. Thank you very much for your attention.